Sharing my screen. You should see my screen now. Today in the lab what we're going to be doing is discussing and starting the Unknown Project. Can you guys hear me okay?
Yes.
Okay. We will have the first half hour on the lecture on Chapter 5, but then we'll discuss and start the Unknown Project. Any question about any of that? All right, so let's begin. Chapter 5, Microbial Metabolism. Always, I give an introductory slide giving you the major goals and a rough outline. Know the terms, which catabolism, anabolism, activation energy, enzyme, active site, competitive inhibition, non-competitive inhibition, also called allosteric inhibition, redox reactions, that's oxidation reduction, what are photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. We're going to talk primarily about aerobic respiration. We'll then discuss its steps, how it differs from anaerobic respiration, and then we'll discuss briefly anaerobic respiration, and then go on to talk about fermentation, and then lastly, cyclic and non-cyclic photosynthesis. We should come to understand what feedback inhibition is. And of course, you should know aerobic respiration in detail because we will discuss it today. Well, in this lesson, we'll only get started today. You should know that metabolism is all of the chemical reactions occurring in a cell. And since we're talking about microbes, metabolism is all the chemical reactions occurring in an organism, because a cell is an organism for the microbes we're talking about. You can separate metabolism into two parts, catabolism and anabolism. Catabolism is the decomposition reactions where you take a large molecule like starch and you break it into a simple molecule like glucose. This releases energy. The energy that was stored in the starch will be released. And some of that energy can be used to make ATP. Let me state that our cells are not good at taking energy released from catabolism and then storing that as ATP. Our cells, I'm talking about human cells, can only do this in two chemical reactions, and that is aerobic respiration, which is a process we'll talk about, and then fermentation, another process. All other catabolic reactions do not generate ATP. So is that clear? So this graph is a little bit of a misnomer, and that is, in humans, most of the catabolic reactions do not generate ATP. Only 2 of them generate ATP. And even in green plants, which are much more efficient than us, and have much more reactions where they can make ATP, the majority of the catabolic reactions do not make ATP. When you convert the energy in one form to another, like the energy in starch to, the energy in ATP and the energy in glucose, there's always energy lost as heat when you convert energy from one form to another. So heat always comes out, and heat is a form of energy. It's just not very useful because the cells can't use it to do work. Catabolic reactions are energy releasing, meaning the energy of starch has a lot more energy in it than the energy stored in glucose, and so to break starch into glucose, energy is given off. Any questions about that? Anabolism is the reverse, where you take simple, smaller molecules and you hook them together to make larger, complex molecules. Anabolism is biosynthesis reactions, and for them to go, you need energy to come into the system to make the larger molecule. That energy usually comes from the energy in ATP, like when we are taking glucose, like if you had a, I don't know, candy bar or something, and then you're going to make it into starch, or not glycocalyx, what is that? Glycogen. You will convert the glucose into the more complex carbohydrate, but that takes energy, and your cells will get that energy from ATP. When you convert energy in glucose to the energy in starch, and energy comes into the system, some energy is lost as heat. And that's because whenever energy is converted from one form to another, some energy is converted into heat. And you don't need to know that, but that's just simply the second law of thermodynamics. All right, any questions about metabolism? You should understand that a metabolic pathway is where one reaction converting one substrate or reactant to a product, that you can convert that product to another product by linking another reaction, and then linking the reactions together so that you get an end product that is made from the reactant, and it goes through several intermediate by a reaction. So this would be one reaction followed by another reaction followed by another reaction. You can link these reactions together to get a metabolic pathway. And metabolic pathways are a sequence of enzymatically catalyzed chemical reactions in cells, where one molecule is converted to another, and how they're converted in the cell is by an enzyme converting molecule A to molecule B. Any question about any of that? The last molecule is important, and we call it the end product. The starting molecule is important, and that's either the starting substrate or the reactant. All of the intermediates are a product for this one reaction, like B is a product of this one reaction, but it's an intermediate that then becomes the reactant of this reaction. Anyway, cells, to get specific molecules made, they can make a metabolic pathway to make that molecule. In cells, there's a huge amount of information, and this is trying to show you some of the information that is going on within a cell, and you only need to memorize all of it. Only kidding. Cells can create a molecule from a reaction by the collision theory. The collision theory states that chemical reactions occur when atoms, ions, and molecules collide with each other. And if they have enough energy, the reaction can proceed, meaning the two molecules colliding with each other, if they have enough energy, they will make the product. The activation energy is the energy needed to disrupt the electronic configurations in the reactant and then make the product. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. You can increase the reaction rate by increasing the abundance of enzymes, and then, let's see, the reactant is more likely to find the enzyme is more likely to convert the reactant into the product. You can also increase the reaction rate by increasing the temperature. When you increase the temperature, the molecules move around with more energy, and because they're moving more, they're more likely to collide, and if they collide, they also have more energy, and so you're more likely to get the reaction to proceed. If you increase the pressure, that does the same as increasing the temperature, and that's not something that's intuitive, but if you increase the pressure, you do increase the reaction rate. I suppose that's how diamond gets made. That is, it's under pressure and heat, and because of the pressure, you get the diamond form in the reaction from carbon or coal. Enzymes reduce the activation energy of a chemical reaction. For example, if you have the reactant A and B, if you give it enough energy, it can convert into the products A and B. For example, glucose right here can be converted into CO2 and water, but to do that, you have to heat the glucose to the activation energy without the enzyme. And how you can do that is you take a blowtorch to the glucose and you'll cause it to burn into carbon dioxide and water. Energy is given off by the process, and that's why the glucose, or the initial product, is higher than the energy in the glucose in the water. Energy is given off, but to go from the reactant to the product, you have to go over the energy hump, and that would be the reaction without an enzyme. And that's a very high energy hump. If you don't know, a blowtorch is very, very hot. Okay? And yet, in the room you're in, glucose can be converted into CO2 and water all the time. And where is that happening? Hello? Where is it happening in the room you're in where glucose is converted into CO2 and water all the time?
Breathing.
Say again?
Breathing or respiration.
Oh, yeah. From aerobic respiration in your cells is what I was looking for. And that is glucose can be converted into CO2 and water. And does that take the temperature of a blowtorch? That's yes or no. Well, you guys really do not like cooperating on the lesson. The answer is no. That's happening in your cells at 37 degrees, and that we call the reaction activation energy with an enzyme. And that's much lower a temperature than converting glucose into CO2 and water with a blowtorch. So 37 degrees is much less than something like 1,000 degrees. Okay? So the activation energy with an enzyme is much less, not shown here, but much less than the activation energy without an enzyme. Any question about any of that? That is why enzymes can allow chemical reactions to occur in your cells at your body temperature, and then the reaction happens quickly with the enzyme. Enzymes are simply biological catalysts. A catalyst, all it does is to speed up a chemical reaction. You don't need to know how many times, but the catalysts and enzymes speed up chemical reactions 10 to the 8th to 10 to the 10th times. And so that would be around 1,000,000,000 times. That's 10 to the 9th. Enzymes are encoded by genes, so the chemical reactions occurring in your cell are encoded by the information in your DNA. You should know that enzymes are specific for a chemical reaction. Generally speaking, one enzyme will work on one molecule to perform one chemical reaction. Now, for our purposes, that's good enough. You should know that occasionally you can get other molecules that can react with an enzyme. And we have certain drugs, for example, that act by binding to and interacting with the enzyme. Okay? But generally speaking, we'll say an enzyme is specific for one molecule and can perform only one chemical reaction. That specificity is due to the three-dimensional shape and the folding pattern of the enzyme. Each enzyme acts specifically on the one region of the specific substrate, and I'll show you that in a little bit. You should also know enzymes are not used up in a chemical reaction, so after the chemical reaction is done, the enzyme is unchanged, and then it can bind to another reactant and perform the reaction time and time again. And that's why enzymes can speed up a chemical reaction. How they do this is an enzyme forms a enzyme substrate complex, and that puts stress on the bonds of the chemical reaction, which can make it more easy to convert the bonds of the chemical reaction to convert the reactant into the product. And that also is responsible for lowering the activation energy with an enzyme. You should know that most enzymes end in the name ACE or ASE. It isn't true for all enzymes, like renin. You've probably heard of that enzyme in your stomach. It helps digest milk and it curdles milk. That is an enzyme that was around before the convention that we give enzymes the name ACE, but most enzymes end in ACE, and we will talk about ligase and catalase, but you don't need to know any of the enzyme names. Okay? You should know that enzymes may not work in a vacuum. Some of them can work in a vacuum, and all they are is an enzyme, like a protein portion that then will react to the substrate. But other enzymes require a coenzyme or a cofactor. And these are helpers that bind to the enzyme and then allow the substrate to bind to the complete enzyme. And you don't need to know the names, like apoenzyme and haloenzyme, but you should realize that some enzymes have a coenzyme or a cofactor. A cofactor is simply a element like iron, zinc, magnesium, which helps the enzyme function. A coenzyme, on the other hand, is a biological molecule which helps the enzyme function. There are a number of important coenzymes. You only need to know the first two in this table. NAD or NAD+, nicotinamide adenine dinucleotide. It's involved primarily in catabolic reactions, and we'll talk about it when we're talking about aerobic respiration. FAD or flavine adenine dinucleotide is also involved in aerobic respiration, and that's why you need to know these two, because we'll talk about them in aerobic respiration. The others we're not going to talk about or talk about much, and they are just other important coenzymes. And I won't ask you questions about the other coenzymes. But NAD and FAD are fair games because we're going to talk about them when we talk about aerobic respiration, which will be on our next day, on Thursday. All right, any question about any of that? All right, if not, this is showing you the mechanism of enzymatic action, where an enzyme binds specifically to a substrate, and the enzyme binds to a substrate in its active site. The portion of the enzyme that binds to the substrate is called the active site. And the two complex together, and that puts stress on the substrate, converting the substrate into the product. Once the product is made, it then falls off of the enzyme, and then the enzyme can rebind to another substrate. This is called the lock and key model, where one enzyme fits one substrate, like one key specifically fits one lock. Any question about any of that? Now this picture is showing you a more realistic model of how an enzyme acts. And this we would call the hand in the glove model. I'll explain that in a minute. Where one enzyme, shown in blue, binds to one substrate, shown into red, and then they form a complex before the substrate is converted into the product. And what do you notice about the substrate and the enzyme? There's actually three things to notice.
We fit together well.
Yeah, they fit together in the active site of the enzyme. What else do you notice? Can you repeat that? That was so lightly said I couldn't hear it.
The substrate changes state.
The substrate changes state. That's very observant of you. The substrate, if you look at it really closely, you'll notice that its shape is slightly changed. It's a little flatter here. And that's what's happening when the substrate binds to the enzyme. It puts stress on the chemical bonds of the substrate, changing the substrate into the product. So it's very observant. Most students never see that. And then the third thing is that the enzyme is much larger than the substrate, and the enzyme changes its shape when the two are bound. And so this is why it's called the hand in the glove model. The glove, or the enzyme, changes its shape to mold to the substrate. And I would say that the two are actually beyond that. It's more like a shapeshifter's hand fitting into the glove, where the glove is molding to the shape of the hand, but the shapeshifter's hand is also changing its shape to fit the glove. And that's what's happening in an enzymatic reaction, where the substrate and the enzyme are both changing their shape or structure to form a complex with each other. and then convert the substrate into the product. All right. Any questions about that? So you can talk about the lock and key model, and it's good when you're talking about one enzyme binds to one substrate. But you can talk about the hand in the glove model, and even better, the shapeshifter's hand in the glove model. And if you know Star Trek Deep Space Nine, you know what I'm talking about when I'm talking about a shapeshifter. That's somebody who can change the shape of their body. Any questions about any of that? All right. Let's go on and talk about there are different factors that can influence the activity of an enzyme. Enzymes don't work in a vacuum. They have to work at the correct temperature, the correct pH, the correct salinity, the correct heavy metals. Generally, they work without alcohols, without UV radiation, because all of these can cause the protein of an enzyme to denature. And if the protein denatures, the enzyme will not function, at least at the optimal rate, if it functions at all. There are a few other factors that can influence the activity of an enzyme. You can increase the substrate concentration, and that can speed up a chemical reaction. You can also affect the enzyme's activity with the occurrence of poisons, like cyanide or fluoride, can destroy enzymes, and that will obviously stop the enzyme activity. You can inhibit an enzyme, and there's two types of inhibition. We'll talk about them next time. There's competitive inhibition and non-competitive inhibition. And non-competitive inhibition is frequently called allosteric inhibition. So those two mean the same. And also there's feedback inhibition, which we'll talk about next time. Any questions on any of this part? Factors influencing enzyme activity? If not, I'm going to stop here and then continue with the lab. Let me see if we got far enough in Chapter 5. We might be a little behind schedule, but we'll try and go quickly in the remainder of this lesson. Yeah, it's a pretty long lesson. All right, any questions before we move to the lab? Okay. If not, in the unknown report, if you can find week 5 of the lab, I've got a bunch of information about the unknown project, and you should have read the unknown instructions, because I'm going to talk about that now. I've got a little example of an unknown report, and this was made by an instructor many years ago. I think it was 2004. So it'll be a little different because this is actually a report that somebody wrote up when they went into the lab and did the report. Yep, Travis Kubota was a microbiology instructor back in 2004, and he wrote up this report. So it'll be a little different, especially the start. In the lab, we normally have students, we give them a mixed culture, and they got to streak it out for colony isolation 2 times and get a pure culture and then do a gram stain. And you guys are going to be given a pure culture at the start, but you will want to quote online streak it out, meaning what you're saying, you're streaking it out on day one, and you're given a pure culture, but you want to doubly make sure it's pure, and then establish your stock culture from one single colony. And so you can do that. It's going to be all done online, and I call that a virtual project where all the work is done online. The point is that the example is a little bit different in the start, but then your example should follow the report that Travis Kubota made for you guys. Let me back up again. I've also got a link for building a dichotomous game plan. This project, your unknown project, should be using a dichotomous game plan where you do a test which has two possible results. You get that result. I can't find it. There it is. You get the result. It's rectangular, so we know it's species A and B. It's not species C and D. And then you do another test which separates out species A and B. Okay? This is dichotomous logic, where you're separating your possible species into two groups by one test, and then once you get that, you then separate your next into two groups by one test, and if you needed to do it again, you separate this group into two possible results by one test. So in this project, you're supposed to be using dichotomous logic, and that means you're doing one test at a time. Any questions about any of that? And so there's a little paper on using dichotomous logic. If you do not use dichotomous logic to do your report, then you will be penalized. There's a link for the unknown species list and all the possible tests you can run. And I should probably download this because it would be hard to see in Canvas. On the top, we have all the tests you can run, and on the side, we have all the possible species. And here, this is a better way to view it. So, what is there? About 40 different bacteria species. You have to narrow it down. And something that's fast in narrowing it down is if you do the gram stain, you divide the number of species into two. All the top species above the double line are the gram-negative cells, and all the bottom species are the gram-positive cells. And then you can run other tests to narrow down your species. For example, the PR lactose of the gram-negatives will separate them into these species, those which are PR lactose negative, and then these species which are PR lactose AG. A and G stands for acid, and gas are produced. Okay, and how you read this table is there's a key down here. Well, I thought there was a key. Oh, there it is, right here. So the star means there's a specific note about that. V means the result is variable. Variable means that it can be either plus or negative. Okay? So if you run that test and it says variable, that means you have to consider it being both positive and negative. ND means not determined. You treat that like a variable. Where we have an ND, it means this test, like nitrate test right here, we don't use it on these species of cockeye, we don't use this test to determine what species it is. We just don't even run these tests. And that's why it's not determined. All right, any question about any of that? Most of the table, plus and minus, positive result, negative result. A for acid, AG for acid and gas, V for variable. For hemolysis, there's alpha hemolysis, beta hemolysis, and then negative for no hemolysis. I think that's all of the table. There are some notes down here for how to properly read the gram stain, how to properly get the cell arrangement, and I will state that you cannot get the cell arrangement from doing a slide or a specimen from cells that were growing in solid culture. To get the cellular arrangement, you normally have to grow the cells in a liquid broth culture and then stain them. So the gram stain, you cannot determine the cell arrangement because the cells will be growing in a solid culture. And so their cell arrangement may not be correct. When you grow cells on a solid culture, the cells tend to stick together and just clump. All right, you can read about all the other tests. So to get the cellular arrangement, you have to ask for that test. And where did we, there it is there. All right, any question about any of that? Let's back up. I might have that there. The unknown notes is someplace where you will put all of your information. That will be all of your notes, and you'll have a link here that will, let's see, is that upload a page? You'll upload a document, and in that document you'll have your notes that could include your observation, the cell was round. It should include your observation, the cell was round, and then your conclusion, the cell is a cockeye. You should also have your test requests. So at the bottom of the document, you should say, all right, my gram stain was, I don't know, gram positive. The next test I want to run is this, and list out the test at the bottom of the page. You should put your notes in there, meaning what your thoughts are, and you should put in there the species that you're considering. Like of the gram positives, you should say, I narrowed the bacteria down to the gram positive, and it's all the gram positive. When it's something like that, you don't need to list the name of all of them. You can say, my species is the gram positives, and that's all of these here, which I can't highlight it for some reason. Well, we'll do the gram negatives. Nope, I can't do that either. All right, you get the point, hopefully. But later on, you should mention all of the species that were under consideration. Like, for example, after you've done the PR lactose test, you should state, I can't read this, let me see if I can blow it up. Let me see if I can open it again. Nope, can't open it. All right. You should state the species which were AG positive, and I remember that's Citrobacter furendi, Enterobacter something, Enterobacter cloaceae, and I think E. coli. Okay, so you should state the 4 species which are PR lactose AG, PR lactose AG. All right, any question about any of that? So all of that should be in the PR notes, or the unknown notes, and you'll have a link there for uploading the pages. Please do not put your comments and notes in the comments. I will put the comments in the comment section, and when you ask for a test, I will give you the results in the comment section. If you use the comment section, it will be humongous, because the comment section, it's smaller than this, okay? It's only about this long, and so you can only put a few words in the comment. And if you use the comment section, it will make this page run on forever, okay? So do not put your comments in the comments. If you want to put a comment in your paper, put it in your unknown document and then upload the document. You can have almost an infinite number of documents uploaded to your unknown notes page. Okay? Any question about any of that?
Should we do daily notes uploaded separately?
Can you get a little closer to your computer? I'm having a hard time hearing you.
Should we upload our notes every day that we do tests, or do we wait to submit it all together?
You should do it as you're doing the project. It won't be every day, but it will be every day that you're working on your unknown notes project. Okay? You should upload a page every day you're working on your unknown notes project. And that will be a couple of times a week, maybe 2, maybe as many as 3 or 4 times a week. But no, you don't need to do it daily, and you should do it more than once. You should put your results in there, meaning when I give you the results, you should put the observations and then your conclusions in your unknown notes page. All right, so I tell you what to do. And everything is dated. You don't need to worry about dating it. All right, that's the unknown notes page. Oh, and there's a rubric I made for grading the project. Oh, come on. It looks like I might need to upload a new one. Nope, that's correct. It's out of 75 points, so this is the correct grading rubric. I will figure out if all the relevant tests were used, were the correct species included at each step, is the report complete, is there effective and clear communication, was this correct species identified at the end, and that'll be a total of 75 points. There are certain parts that have to make the report complete, and let me talk about what you need to do to get the report complete. I might have already opened that, but we'll open it again. Nope, that's the wrong one. Oh gosh. I thought I had it here. Oh, there it is. All right, this is a 75-point lab project. You'll be working on it from now until almost the end of the term, where I'm going to give you an unknown bacteria. Because this is going to be done virtually, all I'm going to do is give you a number that's going to represent an unknown bacteria. And then you're going to be running specific tests to try and identify that unknown bacteria. So you'll request online in your unknown notes page a test, and I will give you the results after that test is incubated, which will be the results for that test. And let me talk about how you can find out how long the test needs to incubate. Do you need that any further? Oh, I think I'll keep that just for a little longer. Under the week 5 lab, I've got the unknown tests, week 5 lab unknown tests. These are all the tests you can run to help identify, oops, you don't have that seen. Now you got it seen. Make sure that one's up there. All right. These are all the tests that you can run to help identify the species. So 6.5% salt, 7% salt, bile-esculin, those are 2 tests, by the way. Bile-esculin, the capsule stain, catalase, etc. You can run all these tests. And when you ask for a test, you should look at it. Let's do the bile-esculin. To see what that test involves, what the results can be, and then how long the test has to incubate. So this test runs for at least 48 to 72 hours, although bacilli can be incubated for 24 hours to 48 hours. So this one generally will incubate for about 48 hours before you'll get the test results. So when you request a test, that test has to incubate, and this is how long it will incubate. The longest test takes a week to incubate, and that is gelatin hydrolysis. MR is another long one. The methyl-red MR test takes 5 days to run, incubate, and most of the other tests take 2 days to run. There are a few which are immediate, so that when you request the test, I may send you the results on that day. The catalase is an example, that if you have the cells already established from a previous test, I can give you the results that day. If you don't have the cells from another test, then you need to start growing the cells, and that will take a day, so you'll get the results in a day. All right? Any questions about how long the tests need to incubate? And that you can tell from the, that doesn't back up, from the different test. All right, where was I? Talking about the unknown project. So all of this is going to be done online. Let me state that this would take, if there's 20 students, and on average the student is going to need 5 tests to identify their bacteria species, that would mean that this project would take at least 100 separate communications from me and my 20 students to do this project. And I decided that that's a bit much for one project. So if we were to do this all by email, which we're not doing it, we're doing it on the unknown notes, but it's similar. I've got to go into the page and make a response, and so there will be around 100 different requests and 100 different responses. That's a lot of work on my part. All right, so I decided that initially I'm going to choose the tests that you're going to run. And you're going to follow dichotomous logic, but you're going to start, let's see, you're going to start with the gelatin hydrolysis test. If I get the results out today, it'll start today. If And you're going to be doing that. And then if I start today is Tuesday, so let me think. 2 days, so on Sunday you'll start the gram stain, but you won't look at the cells until Tuesday. And then on Monday you'll start the PR lactose test. And so you're following dichotomous logic. You're running one test, and then running another test, and then running a third test. But instead of my emailing each of you 3 times, so that would be like 60, is that right? 20 times 3, yeah, 60 different emails. I'm going to put it all together and email the entire class. Then you'll have your unknown number, and you'll find your unknown number and find what your results are for that number. And instead of having 60 contacts, I'm going to only have 1. And that will greatly reduce the scope and the burden of this project, at least on my end. Okay? It'll speed things up for you, and then you'll have the species narrowed so that you'll have an easier time determining which test to run. Any question about any of that? Now, the first time I send you the information, it'll be in an email. After that time, all work should be done in your unknown notes pages, and all results will be done in your unknown notes page. If you email me that you would like a test, I'm going to reply back to please put that request in your unknown notes page, that I'm not going to honor a test request in an email. Any questions about that? So put it in your unknown notes page. And there is a reason for that, and that is I want all the information together so that if either you or I want to look it up, we can easily find it in your unknown notes pages. Any question about that? If we were to do that by email, it would be almost impossible for me to find that information. All right, so this project should use dichotomous logic. Shotgun logic should not be used and will result in a loss in points. You should make daily lab notes whenever you're working on the project. You should make your notes and then submit the document to your unknown notes pages. This is a little bit out of date. It's when we actually did work in the lab. I didn't correct it here, but it's corrected at the top. The unknown project is worth 75 points. Of course, it has to be typewritten. Please use 12-point font. As you notice, I've got glasses. Anything less than 12-point font will be hard for me to read. I'm not going to dock you if you don't use single space, but it should be single space. It should be in bullet font, but if you like to use regular paragraph writing, that's fine too. You should put in your project your descriptions of the test you run, the information you hope to gain from the test, and then the observations you made from those tests. You should also put below your observation your conclusions. An observation is what you see. The cell was round. The conclusion is what you interpret from that, and that is my cell was gram-positive cockeyed. Okay? So I should say your cell was purple and round. And the conclusion is the cells were gram-positive cockeyed. You may include photos, tables, I will give you photographs of the results, and if you include it, it's helpful, but it's not required. You must have one table or a flow chart at the end, which is going to give you how you identified your project. And I'll talk about that in a little bit. But you should have a table or a flow chart going through the logic you use to identify the species that you think it is. You don't need to ever write in the details of the test. All you need to do is say, I ran the gram stain, and here are my results. Okay? I already know the steps of the gram stain, so you don't need to elaborate on the steps of any of the tests. You should give your thoughts and words why you're running a test and what you hope to achieve, and what species you hope to narrow it down to, or what species are under consideration. All of the work should be in your own words, and if you use from another source, you should put it in quotes and say where you got that information from. And there's the part about the table at the very end, or the near the end, outlining how you identified your unknown. I'll give you an example of that in a minute. You should also have a microorganism paragraph, meaning if you think it's E. coli, then you should have a paragraph giving a description of E. coli. And that can be just anything of importance about the diseases E. coli causes, the modes of transmission, common sources, where E. coli is found, the treatments for the disease, and any other useful information. The microorganism paragraph should not be longer than 1 or 2 paragraphs. Do not exceed a single page for that. The project itself should only be 5 or 6 pages, not counting your references, which can be an additional page. You should credit any sources of information you got that's not known, and you can use any literature citation you want, like APA or MLA. Just be consistent and always use one citation format for your document. I mean, you don't mix your formats. Okay, here's an example table which shows the first test the student did was the gram stain, and they narrowed it down to these possible species with the gram stain. And then they did the catalase test, and that narrowed it down to 4 species. They did the PR mannitol test, and that narrowed it to 3 species. They did the PR glucose test and narrowed it to 2 species. And then they did the citrate test, and it narrowed it down to 1 species. And then they wrote their lab report and the paragraph about Micrococcus varians. Okay? Any question about any of that? So you must include a table or a flow chart which gives you the process of how you identified your unknown. No plagiarism is allowed. And we've already talked about the grading criteria. And this talks about it a little bit more in greater detail. I'll let you read about this. This is what the lab would do if you actually go into a lab. We'd give you a mixed culture of 2 species, and then you grow it on these plates, separate it out onto a single colony, and then streak that out onto another plate to get a second colony isolation, and then grow up your stock culture from a pure culture that's gone through 2 colony streaking events and then you do your tests, okay? But you won't be doing this. I will give you one pure species, and it's true you will online say you're streaking it out to grow up your stock culture, but that's all you're going to be doing, okay? And if I give you that information today, you could start that, but we'll see if I can get the information out to you tonight, okay? I'll try and get out the unknown number and start the the glucose hydrolysis test. This is showing you an example of dichotomous logic. If you need more information on dichotomous logic, you can go through it. You do not need to answer these questions for the project. Occasionally students see this and they think I want these answers. I do not want these answers. Okay, this is just to help you understand dichotomous logic. All right, any questions about the instructions? Any questions about the unknown project? If not, I think we're done.
Question for you. If we do have questions for you in regards to the unknown, can we email you there, or does it need to all stay in the notes?
If it's a question about the test, you should put it in the unknown notes. If it's a general question about the project, you may email me. And if you get that confused, I will respond to a question in the emails, okay? But generally, if it's specifically about the project, I do want it to be in the unknown notes page. All right?
Okay, thank you.
All right, any other questions?
Thank
All
you.
right, I will see you on Thursday.
Thank you.
All right.